Hey everyone, my name is Jenna Kluke and today I'm going to be doing Lab 3B. I will be using a chemical and physical method to be able to control microbial growth using my leftover 21-day sauerkraut brine. I have all my PPE on, I have my safety glasses, my hair is up, I have my lab coat on, and I have gloves. I have already disinfected my working surfaces with my 10-to-1 bleach solution. So let's get started. For this experiment, I prepared 3 samples. My first sample is the untreated 21-day sauerkraut brine, which serves as my control. This sample was not exposed to any treatment and represents the normal microbial growth present in fermented brine. I will compare my treated samples to this control after incubation to determine how effective each treatment was at controlling microbial growth. My hypothesis is that both the physical and chemical treatments will reduce bacterial growth compared to the untreated control. However, I expect the alcohol-treated sample to show the greatest reduction in bacterial growth because alcohol rapidly disrupts cell membranes and denatures proteins. I also expect boiling to significantly reduce microbial growth because moist heat denatures proteins and damages essential cellular structures within microorganisms. For my physical control method, I selected moist heat boiling for the sauerkraut brine. I have measured about 15 milliliters of my 21-day brine and I'm adding it to a small saucepan. I'm going to bring the brine to a rolling boil, and once it's reached the rolling boil, I'm going to allow it to boil for about 1 minute. Then I'll remove it from heat and allow it to cool completely to room temperature before plating. Turning on the stove. Heat works by denaturing proteins, which causes the proteins inside microbial cells to lose their normal structure and function. Heat also damages enzymes and other essential cellular components that microorganisms need to survive. While boiling does not reliably destroy bacterial endospores, it kills most vegetative bacterial cells and should greatly reduce the number of living microorganisms in the sample. As you can see, it's starting to boil, so I'm going to let it boil for a minute and I'll be back once it's cooling. So once the brine got to a rolling boil, I allowed it to boil for about 1 minute and then I removed it from heat, and now I'm letting it cool completely to room temperature before I plate it on my agar plates. While my physical control method cools, I'm going to start my chemical control method. I selected 70% isopropyl alcohol. Alcohol is classified as a disinfectant and is one of the chemical control methods discussed in our laboratory manual. Alcohol works by disrupting microbial cell membranes and denaturing proteins. Once the cell membrane is damaged, the contents of the cell begin to leak out, causing the microorganism to die. A concentration of 70% alcohol is considered more effective than higher concentrations because of the presence of water helps the alcohol penetrate the microbial cell more effectively. To perform this treatment, I already measured out 10 milliliters of my 21-day brine, and I'm going to add 10 milliliters of isopropyl alcohol to my mixture, and I'm going to gently mix the solution and allow it to sit for 5 minutes so the alcohol has sufficient contact time to act on the microorganism. Everything used in this lab has been disinfected or sterilized or brand new out of the packaging. And this is 10 milliliters. I'm going to gently mix it. And I'm going to allow it to sit, and I'll come back and plate it in 5 minutes. Now that both my physical method has been cooled and I've waited the 5 minutes for my chemical method, I'm going to put them on a slide. So my physical, which was my boiling, is going to go on the left side. I'm going to try to get this bubble out of there. And this is using a brand new sterile pipette. And I'm going to get a sample from my chemical. I'm going to add a drop to the slide. My slide has been labeled properly, and I am going to let these completely dry before I gram stain them. And now I will be doing my EMB plates and TSA agar plates. I'm going to do my plating for my TSA and my EMB plates. They have been properly labeled with chemical and physical on the side. I put big C's and P's on them just as a quick reference. I'm going to have them taped shut, and I'll come back in 24 and 48 hours to show you what the results are. After completing both treatments, I had 4 agar plates ready for incubation. My boiled sample was streaked onto 1 TSA agar plate and 1 EMB agar plate. My alcohol treated sample was streaked onto 1 TSA agar plate and 1 EMB plate. Each plate was clearly labeled with the treatment type, date, time, my initials, and the plates will now be incubated at room temperature, and they will be checked at 24 hours and 48 hours, and I will evaluate the bacterial growth at both. My control samples are already done, and I will add those into the results at the end. After incubating the plates for approximately 48 hours, I compared both of my treated samples to the untreated 21-day sauerkraut brine control. I'll start with my physical control method first, which was boiling the brine for 10 minutes. On the Nutrient Agar plate, no bacterial growth was observed after 48 hours of incubation. This suggests that boiling was effective at eliminating the microorganisms that were able to grow on this medium. On the EMB agar plate, there was a small amount of bacterial growth present. Compared to my untreated control, there was noticeably fewer colonies, indicating that the boiling treatment significantly reduced microbial growth but did not completely eliminate all bacteria. This result is expected because boiling is very effective at killing vegetative bacterial colonies, but it may not destroy all heat-resistant microorganisms or bacterial endospores. When I examined the boiled sample using a gram stain, I observed a significant reduction in the overall number of bacteria compared to the control sample. There were considerably fewer gram-positive cocci, while gram-negative rods were still present. Although gram-negative rods remained the predominant cell type, there were fewer of them than observed in the untreated control. These observations indicate that boiling greatly reduced the bacterial population, although did not completely eliminate every microorganism present. Next is my chemical control method, which I use 70% isopropyl alcohol. After 48 hours of incubation, both the Nutrient Agar and EMB plates showed only a few bacterial colonies. On the Nutrient Agar plate, the colonies were pinpoint in size, cream-colored, opaque, and slightly raised. The EMB agar plate showed slightly more colonies than the Nutrient Agar plate, but they were still very limited in number. The colonies on the EMB plate were clear, opaque, pinpoint, and slightly raised. When I performed the gram stain on the alcohol-treated sample, I observed the fewest bacteria of all the samples examined. There were subsequently fewer gram-positive cocci than present in both the untreated control and the boiled sample. The remaining bacterial population consisted primarily of gram-negative rods, but the overall number of bacteria was much lower than either of the other samples. Overall, both treatment methods successfully reduced microbial growth compared to the untreated control. However, the 70% isopropyl alcohol treatment was most effective, as it produced the fewest bacterial colonies on the agar plates and the greatest reduction in bacteria observed during gram staining. These results supported my original hypothesis that both methods would reduce microbial growth, with the alcohol treatment producing the greatest overall reduction. Thank you for watching.